Today I'm presenting about more of a research project and data analysis on the effects of harmful algal blooms on the environment. But before we start, just a little bit about who I am. My name is Sophia Hsu, and I live in the Bay Area, California. I'm currently a junior at Carmel High School. I plan to major something in biology or either neuroscience or genetics. So yeah. Before we start, just as a little background knowledge into what a HAB is. So a harmful algal bloom is a phytoplankton bloom that produces toxins, depletes oxygen in the water when it decomposes, or otherwise disrupts the ecosystem, therefore making it harmful. And this is caused by many things. They could be favorable winds or currents, or after outstanding natural phenomena. But more recently, scientists have speculated that HABs have occurred more frequently due to agricultural runoff, which is basically when the nutrients from the soil of the farmers run into the water. And they are recurring events off the coasts of the US and all over the world. But what makes harmful algal blooms harmful are that they release toxins into the environment. And obviously different algae release different toxins, which makes it a little harder to study. But the main effect of these toxins in the water is known as shellfish poisoning. And this is where toxins accumulate in shellfish, and then when they're passed up the food chain to higher level organisms such as mammals and humans, they cause nausea, vomiting, confusion, and even death because of these toxins. However, the main focus of my research is that these toxins appear to have absolutely no effect or very minimal effect on lower level food chain animals such as like fish and filter feeders, which are clams, mollusks, etc. However, they have severe effects on larger mammals such as sea lions. So that is the main focus of my research. And they also deplete the oxygen levels in the water, but that is not very important to my entire project. So I chose to study two strands of phytoplankton. The first one is Pseudo nitzschia, which produces domoic acid, also known as a red tide. And Pseudo nitzschia is associated with amnesic shellfish poisoning, which affects the brain and the heart of higher level mammals. I also chose, and this is a map of phytoplankton bloom off the coast of California. And the effects of domoic acid on sea lions have been well studied, since scientists have determined that prolonged exposure can lead to effects such as like seizures, infertility, and death. So my hypothesis was, it's already well proven that domoic acid leads to a decrease in sea lion populations. But what I wanted to study was whether this also holds true for lower level organisms such as fish and filter feeders. I also chose to study Alexandrium Catenella, which produces saxitoxin. So Alexandria catenella is associated with paralytic shellfish poisoning, which is a little bit different from domoic acid. But since it blocks nerve receptions and things like sodium pathways in your blood. But this is routinely found off the coast of Alaska and northern Canada, which is where I focus my research. This is a little bit different because Alexandrium catenella can be stored in cysts and then bloom into cells when the temperatures are right. So this makes this even more dangerous because they can go into their resting stage at the bottom of the benthic realm or the seafloor when they're the conditions are not right, but once the conditions are met, that's the nutrients and the temperature, they can continuously bloom, which means that they can stay in the same area for a very long time and cause a lot of harm to the animals that live there. Okay, so to conduct my research, I first of all read many literature reviews. So some sources that I took from were the National Library of Medicine, Science Direct, and also National Association of National Oceanic and Atmospheric Association. And some keywords that I used just to search for these papers were things like the toxins and also shellfish poisoning. For my data analysis, I use publicly available data sets since most of this information is classified. But for domoic acid, there was a large bloom in 2015, which really brought this problem to the attention of humans. This is because of an excess amount of nitrate and silicate in the water. But since domoic acid is also already very well known for causing amnesic shellfish poisoning, I used data from fish from that year that was collected along with same crews as the levels of acid in the water, um, and compared them. I also used a data set by the Arctic Data Center for the Alexandrium Catenella cyst and cell counts in 2018. Since this is also a relatively new, so this is actually the first year that saxitoxin and Alexandrium catenella have been really studied. So unfortunately, there is no other data on this subject, and that's I'll get to it, but that is one of the flaws. So for my findings, I used RStudio and Python to run regressions across all my data, and I ended up with this Spearman rate correlation. As seen from the data, there's a moderately strong positive correlation between the dissolved and particulate domoic acid in the water and also the domoic acid found in various parts of the fish. I believe you can see that right about here and here. However, I think it's important to note that all the fish in this study were caught live, that even with high levels of domoic acid, which is up to about 50,000 parts per million, they had no apparent effects on the fish since, they weren't dead. And I also think it's important to note that a recent study found that levels of domoic acid in shellfish and fish were actually highest during the winter months, despite most algal blooms happening in the summertime. And I believe that this probably indicates that domoic acid can probably be accumulated and stored in lower levels of animals, such as like zooplankton, before moving to a higher trophic level and affecting higher level mammals such as sea lions and sea otters. However, there was a very low correlation found between Alexandrium catenella cell count and the sea lion counts. Some potential problems that could have happened in my data was that I rounded too much when lining up the coordinates. We also had very limited data since this was only from about 2 months out of the 2 years, since this is obviously not a very well studied topic, and also not publicly available. I also used a geocode catch in Python to change the site names into coordinates, so that could have been not that accurate. However, I think another reason that my saxitoxin data could have been inconclusive, because if we look at this map, I studied the southern coast of Alaska and also northern Canada right about here. And if I zoom in a little bit, we can see that, we can see that the red represents the mammals affected by domoic acid in the same area, while the purple also affects represents the mammals affected by saxitoxin. And since they're very mixed around, I think that this could have meant that domoic acid was causing the change in sea lion populations instead of saxitoxin. And as for my conclusions, obviously there's a clear connection between domoic acid in the domoic acid levels in the water and fish. And I think this explains how domoic acid moves up the food chain and into higher trophic levels. And obviously there's evidence that domoic acid can be stored in lower level organisms. I think, unfortunately, the saxitoxin data was inconclusive, but I think we can assume this is either due to bad analysis or, as we saw, the overlapping between the areas of domoic acid and saxitoxin. For my further steps, for domoic acid, I would like to repeat the data analysis for a longer time period so that since that was the data that I've collected was only from about 3 months, and obviously we see that they can go all the way into the winter months. I'll also include other species such as like shellfish and even some sea lions and zooplankton. in saxitoxin, I would also like to compare the sea lion counts to the domoic acid in the area so that we can see if there is a correlation between those two, and that would explain why there is no correlation between the sea lion counts and the saxitoxin. And again, I'd like to include other species. Thanks for listening. Do you have any questions?
Thank you, Sophia. We have time for one quick question.
Sorry.
First of all, I'm terrified by all this, but the question that our audience asks is, I mean, throughout your research, you talked about some next steps, but they're curious if there was anything specific that really surprised you that may not have been in the scope of your research project, but you would love to explore in the future.
I think what I wanted to focus on was more of the biological effects of the toxin as to why it only affects mammals and not things like fish and mollusks, because I kind of want to go into like the nitty-gritty, like microbiology. How does it affect the brain, and is it because mammals have more complex structures? Is that why the toxin seems to affect them more? So that's something that I would also like to study.
Love that. Well, thank you very much. That is fascinating and terrifying.
Yeah.